discuss the basics regarding the molecular biology. They should be able to classify normal regulatory genes, and they should be able to discuss in detail the important proto-oncogenes and tumor suppressor genes. Now, yes, it's all about genes, the molecules, DNA, and the genetic mutations in this chapter. So before going ahead, we should know the normal physiological process that occur in a cell during the multiplication or the growth phase. So we have the cell cycle, which has multiple phases during which it completes its division or proliferation. The first one is G1 phase. The G1 phase starts as the it comprises of the growth of the cell, multiplication of the cell. S phase is actually the synthesis phase, in which there is DNA synthesis, and after which there comes the G2 phase, which is the growth and preparation. The cell is being prepared for mitosis of the next cell division. And the M stands for mitosis, that is the cell division, exact cell division occurs in this phase. So there are basically four phases of cells. Why we need to understand the phases of cells? Because the nature has given different checkpoints at different levels within the cell cycle itself. So that if there is an error within the cell, or error in the chromosome, or a defect in the DNA, or any other environmental effect that is affecting the DNA or the chromosome component, that is being naturally checked by these checkpoints. For example, you can see that since the cycle starts from G1 phase, and then at the point of entering of the cell, which is a growth phase, and from the entering of point of cell from G1 to synthesis phase, from growth phase to synthesis phase, there is this G1 checkpoint, which sees that, which checks that whether the environment is favorable for entering into the DNA replication phase. Again, when it goes into the into the G2 phase to the M phase, at this there is G2 checkpoint, which checks that since the DNA has been replicated, but is all DNA replicated, and is the environment is favorable to enter into the mitotic phase. And then at the and if everything is all right, the cell enters into the mitotic phase. We all know what happens in mitosis. There is cell division occurs. Now, even at the exit of the mitosis phase, that is, even after the mitosis has been finished, when the cell is exiting into, is completing the metaphase, there is a metaphase checkpoint, which shows that are all chromosomes attached to the spindle. Because we know, and you will be studying in the mutation chapter, that if the arrangement of the chromosome is affected, then also there are chances of cells getting acquire new mutations. So these are the natural checkpoints that are being already available in our bodies within each cell. Now, there are certain terminologies that you'll be frequently coming across during the lecture, or during when you will be studying this chapter. So I would ask you to open up the Robbins pathology pocket book for this, because in the main textbook the chapter is too prolonged, and some excess information is also given for postgraduate level students. So I would recommend you to read the short book, so that the short Robbins is also there to give you a good concept. If you listen to this lecture first and open up the book later on, I hope that you won't find much difficulty. And even if you find difficulty, you can give a comment below the lecture tab, and I'll be answering your questions. So terminologies are the germline mutations. Now, what are germline? We all know that the fetus, the babies develop from the germ cell. The eggs are developed from the germ cell. So germline mutations are certain kind of mutations that are passed on to the next generation. So we call them the mutation within the germ cells. Now, there are Other kind of mutations, which are somatic mutations. The somatic mutations are actually it's an alteration within the DNA after the conception. First, you need to understand what is a mutation. Mutation is a phenotypic change or a change in the coding sequence of the DNA that occurs due to multiple factors, due to any of the factors. So any change in the DNA is basically a mutation. Now there are two types of mutation. We just discussed the germline mutation, which occurs within the germ cells, and the somatic mutations, which occurs in all cells except germ cells, that is except sperm and eggs. And it usually occurs after conception, so there are no chances of forwarding this kind of mutation into the next generation or the next offsprings. Now, there is a terminology frequently used in molecular biology, and that is genome instability. Why would a genome? Genome is actually a bigger, broader term than the gene. It has arisen from gene, but what is a genome? It actually is the total genetic makeup of an individual. For example, me or you, we are an individual, and the total genetic makeup of us is known as a genome, right? So why does a genome get instability? Instability is when there are frequent mutations and frequent changes within the DNA structure. So a high frequency of mutation within the genome of a cellular lineage is known as genomic instability. To understand in simple form, it means that a certain kind of individual who has multiple mutations, who has frequent mutations within the DNA, his genome is unstable, and that is known as genomic instability. Now, certain other terminologies are important for you to know. What is senescence? In a cell cycle, we know that there is a cell division, right? So whenever the cell cycle stops, the cell division stops, and if it is a permanent stop, then it is known as senescence. Then there are certain kind of mutations which are known as driver mutations. Driver mutations are a mutation that in which the genes which may lead to cancer, the mutated genes which may lead to cancer formation, they are the driver mutations. Then there are passenger mutations. Driver is, that a driver of a car, and passenger is sitting beside him, right? So the passenger may have mutations, the passenger genes may have mutations, but they will not cause cancer. However, the driver mutations are the mutations in the genes that may lead to carcinogenesis. So they are known as driver mutations. Alright, so that's the difference, and I just explained it to you. Alright, so non-lethal genetic damage lies at the heart of carcinogenesis. So we know that all the carcinogenesis, all the development of cancer, it arises from the mutation within the genetic makeup of a person. So it could be inherited as a germline mutation, or could cancer be a bad luck? There are certain multiple papers published in the literature which can show you that probably cancer is a bad luck also. Some people inherit in their genes at the time of the birth, and when they grow older, they develop cancer. So it could be a bad luck also. So tumor is formed by the clonal. Okay, now we will be talking about the clonal expansion of a single cell. What do we mean by clonal? Let me explain to you in simple terms. A clonal expansion is a single type of cell which is being replicated. Say, for example, that a red blood cell is being multiplied in the body. So a red blood cell, when it multiplies, it only produces a red blood cell. The daughter cell would be a red blood cell. It will not be a WBC, or it will not be a platelet, right? So we call it clonal expansion. That is a single type of clone, right? And the tumors usually display monoclonal proliferation. Monoclonal means they are of one type only. For example, if there is a adenocarcinoma of the colon, then the tumor cell which are being multiplied in the body or within the colon area, they will be of adenocarcinoma type. They will not acquire, squamous type or any other type. So we call it clonal expansion. So tumors are clonal. Remember that. Here, tumor means cancer. In this lecture today, we know that tumors may be benign or tumors may be malignant. But we're talking about malignancy here, the cancer here. So tumors are clonal And the next important point is that tumor progression results from accumulation of mutations. So it is not just a single mutation that is occurring in that individual cell which is causing the tumor, but there is multiplication of all those mutations and then the accumulation of those mutations within the cell lineage. Now, 4 classes of normal regulatory genes occur. The one is the proto-oncogene, that is the growth promoting genes. The second is the tumor suppressor gene, which is the growth inhibiting gene. Then there are genes that regulate the programmed cell death, that is apoptosis. You have learned a good, you have a good amount of knowledge regarding apoptosis. It's a programmed cell death. And then there is a DNA repair genes. So there are four basic kind of genes which occur in this in the carcinogenesis sequence of any type of cancer. Proto-oncogenes are the growth promoting genes. Proto-oncogenes are the growth promoting genes. Now, growth promoting genes are actually they take part, and we will do all of these genes individually. And then there are gain of function mutations that activate the proto-oncogenes. They cause an excessive increase in one or more normal functions. Proto means promoting genes, right? So actually these mutations, they promote the multiplication or further cell division of the mutated genes, right? And that's why they are known as gain of function mutations, because the function, they are overexpressed. The function is gain, that is they are having an excessive multiplication within those genes. Now there are loss of function mutations also, and the mutations that affect the tumor suppressor gene cause a lot of loss of function. Now, what are loss of function mutations? Loss of function means that there are certain kind of genes which display or work as a break in the sequence of the tumor, and those tumor suppressor gene mutations that they actually cause stop in the cell cycle. They try to stop the cell cycle rather than promoting it, and when there is a defect is detected in the DNA or in a chromosomal defect, they try to stop the cell cycle at that particular spot, and that's why they are known as tumor suppressor genes. And because of this function, we call it as a loss of function within the cell cycle pathway. Let me explain to you with this picture. Tumor suppressor gene, the normal function of a tumor suppressor gene is the growth inhibiting protein, right? They have growth inhibiting proteins, so they will keep the cell division under control. Of course, if there are no growth inhibiting proteins, then the cell division will continue. It will just go on and on and on. And we all know that the proliferation could not occur without a certain boundary. I mean, if it crosses that boundary, then how are you supposed to stop that proliferation? For example, that a person is gaining replication of his skin cells every day, but there is a limitation that is being set by the nature to how much of the cell will be multiplying each day. If there is no limit, no tumor suppressor gene role present, how do you expect that the skin will not overgrow? It will not overgrow or develop any tumor formation within the skin. So this is the normal function of the tumor suppressor gene. Now what happens when a tumor suppressor gene is mutated? When there is a tumor suppressor gene which is mutated, there is a defective or non-functioning protein. And this protein is actually what happens, that it will be unable to control the cell division that is occurring in the body, especially of those cells which are mutated. So cell division will not remain under control, and there will be an increased growth in that particular area. Now, apoptosis regulating gene, we all know that in case of an abnormality within the apoptosis regulating gene, then there will be less death of a cell. Less death of a cell means that more survival. So if a DNA defected cell goes into more survival, then you can understand that it will develop into a cancer. What is a mutator phenotype? Now, some cells acquire mutations at a very fast pace. Most of the cells, they resist acquiring mutation, but some cells acquire mutation at an accelerated rate. And those kind of cells, they represent mutator phenotype in that particular case. Now, this picture is important to understand. See here that there is a normal cell, and then there is a carcinogenic, whether it could be a carcinogen coming from virus or coming from an atmosphere. So it's an actually starting a initiating mutation. So this carcinogen induced mutation can the cell can acquire a stem cell like property like a clonal expansion, and then there are a lot of mutations occurring. So a lot of mutations can cause genomic instability, as we just discussed. And then after the genomic instability, these acquire further mutations, that is acquire driver mutations. Driver mutations are those which lead to cancer formation. So acquisition of cancer hallmarks, and then there are additional mutations which occur. So I just told you that cancer is a multi-step process where initially there is only one mutation, but then there is accumulation of more and more mutation, and then eventually the cell or that organ undergoes the cancer acquisition. So mutation we just discussed is mutation is in the coding sequence. There is a change in the coding sequence of the DNA, and it is known as mutation. So coming to the hallmarks of cancer. The hallmarks of cancer are actually the malignant neoplasms. They have several phenotypic attributes referred to as cancer hallmarks. So cancer hallmarks are actually the hallmarks, some of the important aspects of the fundamentals of how a cell changes its own function and how a cell change its morphology. So these are the hallmarks. I'll be trying to explaining to you in simple form. They are not very easy to understand, but I'll try my best. So let's see. These are actually the hallmarks of cancer formation. The first one is sustaining proliferative signal. Proliferative signaling means that a cell continuously divides. It keeps on dividing, and there is no stopping of that. So you know that when there is a mutated cell, with a cell with a mutated DNA, if it keeps on dividing, then how can you expect that cancer will not form? Again, avoiding immune destruction. So there are immune functions in the body, but these cancer cells, these defected DNA cells, mutant cells, they actually avoid this. They skip the immune system, and then they keep on dividing. The third form is evading growth suppressors. Now there are certain growth suppressor hormones in the body, signals in the body, but they do not. The cancer cell do not even respond to that. And then they have a limitless replicative property. That is, they keep on undergoing the mitotic phase of the cell cycle, and they keep on replicating, and nothing can stop them. The another point is tumor promoting inflammation. Now there are certain kind of inflammatory effects which promote tumor formation or cancer formation. Now, invasion and metastasis. These tumor cells, they have the replicating property as well as invasion property. Invasion we have discussed in detail, and metastasis we have discussed in detail. So they acquire these properties within these cells and they invade. Then there is genomic instability. I told you what is genomic instability. It is the acquisition of multiple type of mutations within the cell. And and then of course if a cell or cell lineage is so much unstable, then the cancer is inevitable. Now, inducing angiogenesis. You can see this picture that any of the tumor cell, when it occupies an area, it forms its own blood vessels. They have to survive, they have to live for a longer duration of time. They need nutrition, these cancer cells. So the only way to gain all the nutrients is the blood vessel that will be providing it the nutrition and the metabolism that they require. So they form their own blood vessels, and this is known as angiogenesis. They also resist cell death, and we all know that there are metabolic pathways which are activated only specifically for these cancer cells. So there are so many hallmarks, but we will discuss Briefly, some of them. The first is self-sufficiency in growth signals. I mean, they develop their own growth signals. They do not need any growth signal from the body. So since the growth signal is indigenous, they just keep on multiplying. The second point is insensitivity to growth inhibitory signals. That is, the growth inhibitory signals are present in the body, but these cancer cells, they just skip them. Third point is altered cellular metabolism. What happens is that the tumor cells or the cancer cells, they switch to the aerobic glycolysis. This is known as Warburg effect. And the aerobic glycolysis actually helps them to synthesize their own macromolecules like protein and carbohydrates. So all the nutrition that is provided by them is used for rapid cell growth. Evasion of apoptosis, they skip the apoptosis. They do not respond to anti-apoptotic genes. Then there is they achieve immortality because they have an excessive replicative potential. They keep on multiplying, keep on undergoing mitotic phase. Sustained angiogenesis, as I just explained to you in the picture, they can invade and metastasize, yes, and they can also escape the immune system response against these tumor cells. Now, self-sufficiency in growth signals or oncogenes. Oncogenes are the genes that promote autonomous cell growth in cancer cells. These are known as oncogenes. They are self-sufficient in growth. They do not need any signal in growth. And this is all done by oncogenes. The oncogenes are created by mutations in proto-oncogenes. They encode protein which are known as oncoproteins. So oncoprotein are the proteins of these, the result of these genes, and they are created by mutations. Ability to promote cell growth in absence of normal growth promoting signals. So even if there is normal growth promoting signals not present in the body at that particular time, they do not need any growth signal. These oncogenes, they are able to promote cell growth. Now, this picture can easily tell you the function of a proto-oncogene. What happens in the normal cell when it acquires mutation through any carcinogen, cancer-causing agent, the proto-oncogene is activated and it becomes an oncogene. And this oncogene encodes a protein, oncoprotein, through which it affects the cancer cells, and they keep on multiplying. These cancer cells keep on multiplying without any growth factor signals. Now, this is the list of the signaling pathway that occur, that take part in the oncogenic pathways. You don't need to remember them all, but just remember the name. I'm not going to give you detail of each of this pathway because this is just out of your scope. So, some important pathways are receptor tyrosine kinase pathway, G-protein coupled pathway, JAK-STAT pathway, WNT pathway, Notch pathway, Hedgehog pathway, TGF-beta pathway, and FKB pathway. Now, what are basically the function of a proto-oncogene or oncogene? The proto-oncogenes that actually works the pedal of a bicycle. Imagine that this is the cell cycle, and the proto-oncogenes are the pedals of the cell cycle. What will happen? They will just keep on running and running and running, right? So oncogenes is actually keeps the cell cycle to keep on moving all the time, right? So pro-growth, now there are some growth factors which are self-sufficient in producing their own growth factors. So they form growth factors, they form growth factor receptors, they act in signal transduction through proteins. Then there are some nuclear regulatory proteins which are proto-oncogenes, and then there are some cell cycle regulators. I'll give you a brief touch upon all of these, but not in very detail. So growth factors, some cancer cell acquire the ability to synthesize the same growth factors to which they are responsive. And these growth factors gene itself is usually not altered or mutated. So you can see that there's some, I briefly summarize some important growth factors like platelet-derived growth factor and fibroblast growth factor, tissue growth factor alpha, and hepatocyte growth factor. So you can see that they are PDGF is involved in brain tumor, fibroblast in multiple type of cancers like bladder cancer, stomach cancer, breast cancer. TGF is involved in astrocytoma, and hepatocyte growth factors involved in hepatocellular carcinoma or thyroid cancers. So growth factor receptors. Now certain growth factor receptors are also present in the form of proto-oncogene. These are EGF receptor family, ERBB1 and BB2. And then there are RET proteins, which are, and then there are some KIT proteins, right? So these are the growth factor receptor proteins. And they are growth factor receptors, and they are, you can see that they are involved in multiple kind of tumor formation. This picture can actually explain to you the function of the growth factor and the growth factor receptor. The growth factor receptor is found in the transmembrane area of the membranous area of the cell membrane, and whenever the growth factor comes and attached to it, they actually activate an inactive protein into an active protein. Here the protein is RAS, so RAS is activated. And what does active RAS do? You can see that through multiple processing, it causes activation of the transcription factor, and the transcription factor then obviously takes part in the cell cycle progression and pro-growth signals. This is how cell growth occurs in an individual cell. Now, protein involved in signal transduction. Receptor tyrosine kinases are important, most important in cancer transmembrane protein with an extracellular ligand binding domain and a cytoplasmic tyrosine kinase domain. You have studied the tyrosine kinase pathway in the chapters in physiology, where you did the metabolic pathways, and also in biochemistry, right? So these are exactly the same pathway, but they are more activated within the cancer cells. So the mutant receptor delivers continuous mitogenic signals to these cells. Now this is the tyrosine kinases receptors and non-receptors. The receptors are actually membrane spanning proteins. They live in the membrane, right? They bind to extracellular ligand, and then they cause growth, differentiation factors and hormones. And then the non-receptors are cytosolic proteins. They are mostly coupled to transmembrane proteins, and they cause protein to protein interaction. So coming to this picture, you can see that these are the receptor tyrosine kinase within the transmembrane area, and then they are initially inactive, but when there is a signal dimer binds to them, they become phosphorylated, and when they become phosphorylated, they become active. So the intracellular proteins bind to phosphotyrosine docking sites. Now, signal transduction proteins are many. There are many proteins which take part in signals from the membrane to the cytoplasm, to the nucleus, or to the membrane-bound organelles, right? Like Golgi, like mitochondria. So this transmission signal is very important, but we all know that these transmission signals occur via proteins, the transduction proteins. So these are the type of protein that take part in the signal transmission. That are RAS, BRAF, Notch, JEC2 and ABL. Now here you can see the pathway that these transduction proteins they take up, and is that the pathways that are being taken by these genes, which are involved in signal transduction proteins, they are actually the RAS protein, which is, we will take the example of it, and we will see how the RAS acts within a cell cycle. What is the role? So when you're coming to the growth factor receptors, which we know that they are they lie within the membranous area, the cell membrane area, and here the RAS proteins actually, it has dual function via PI3K AKT pathway and via RAF MEK pathway. It induces proliferation, survival, motility, and angiogenesis. All of these things. So RAS has a signal transduction protein which is multiple factors in different kind of cancer formations. So you can see that there are type of proto-oncogenes which are the growth factor transduction proteins, GTP binding proteins, KRAS, HRAS, NRAS, GNAC, and GNAS. and they are involved in multiple kind of cancers, human cancers involving colon, lung, pancreas, bladder, kidney, melanomas, and then there could be a pituitary adenoma and other endocrine tumors also. The other signal transduction protein is the BRAF, serine threonine kinase pathway. And you can see that in a normal cell, what is the function of the BRAF pathway? It actually transmits the signal from the cell membrane to the nucleus for a normal cell growth. But what if this BRAF is mutated? When there is BRAF mutation, is, for example, in melanoma cells, there are, they are most of the time the BRAF mutation is positive. So what happens is the normal signal is pathway is broken down, and the BRAF itself acquires the the self-replicating activity, and the self-replicating activities gets out of control, of course. So when it gets out of control, instead of formation of two daughter nuclei, they form multiple daughter nuclei, and this is how a tumor size grows. So BRAF point mutations are commonly seen in melanomas, leukemias, and colon carcinomas. Now, then are some non-receptor tyrosine kinase genes also, and they are famous genes which are known as ABL gene. This gene is a proto-oncogene that involves a protein tyrosine kinase involved in a variety of cellular processes. In CML or chronic myelogenous leukemia, this ABL gene is translocated. So there are certain cancers in which the DNA is not mutated, rather there is translocation of the chromosome. In this case, CML or chronic myelogenous leukemia, the chromosome number 9 is translocated to chromosome 22. This picture can show you a very good example of it, and you can see that there is a ABL gene, and then there is a BCR gene, which are normally separated gene. What happens is that a part of an ABL gene is connected to the BCR gene, and you can also see the number 9 chromosome, and it is here, the BCR is at the 22. So what happens is that a part of an ABL is attached with the BCR gene at the 22. So 9 is relocated to the 22 chromosome, and they form a fusion gene, which is a BCR ABL gene. And this BCR ABL gene is a fusion protein that increase the multiplication of the leukemic cells, especially in the case of chronic myelogenous leukemia. Now, there are some nuclear regulatory proteins which are known as transcription factor, because we know that the transcription that occurs in the nucleus. So some of the nuclear important transcription factors which also act as a proto-oncogene, they are MYC, MYB, JUN, FOS, and REL. But you don't need to go into the detail, just remember the name. Now, what is a MYC? MYC activates the expression of many genes involved in the growth. MYC is an important regulating factor, and it is involved in Burkitt lymphoma and neuroblastoma. So coming to this exemplification diagram, you can see that from the outer membrane to the through the RES MYC pathway, this MYC is actually signal transduction, does the signal transduction into the nucleus, and then the nucleus becomes, if it is mutated, then it becomes self-replicative, acquires multiple mutations, and it becomes self-replicated. It becomes a proto-oncogene, does not require further growth signals from outside. Now, there are some certain cell cycle regulators also, especially cyclin D1 is an important regulator of cell cycle. Recall your the background where I told you how many types of stages in the cell cycle, and cyclin D1 is one of the important cell regulator. So overexpression, if the regulator is being overexpressed, like cyclin D1, it definitely is going to cause development of cancer. And it is seen, these kind of overexpression of cyclin D1 is seen in mantle cell lymphoma, multiple myeloma, breast CA, or esophageal cancer. Another cyclin-dependent kinase is the CDK4, and the amplification or mutation is seen in case of glioblastoma, which is a brain tumor, melanoma is a skin tumor, and sarcoma is a soft tissue tumor. So this picture can clearly point out at which level these cell cycle regulator acts. From the G2 to M phase, the cyclin D1 is important. From within the G1 is CDK4 and CDK2, and cyclin A or CDK2 is again in the synthesis phase also. So This is how the cancer mutation occurs also. All right. So what happens? G1 and G2, we all know that G1 checkpoint was for cell cycle, cell size, nutrition, growth factor, and DNA damage. And as we just saw that from the G1 phase, they are CDK4 and CDK2, which are actually the cell cycle regulators in this area. Now coming to the G2 checkpoint, they check for DNA replication and size, and the G2 phase actually is managed by CDK1. Again, mitotic phase is for check for the chromosomal stability, and this is also checked by cyclin D1. So insensitivity to growth inhibition. Now there are growth inhibition pathways and factors that are naturally being placed within a cell cycle, and they are known as tumor suppressor gene. Tumor suppressor gene act as a brake of the cycle. Okay, proto-oncogene was a pedal of the cycle. It keeps on running the cycle, while the tumor suppressor gene, the function of it is to stop the cycle, the cell cycle, whenever there is a defect detected in the cell. Now they actually apply the brakes to cell proliferation, and then abnormalities in these genes. Say for example, a tumor suppressor gene, which is itself a brake, and when a brake of a bicycle fails, what happens? It will just continue, keeps on moving without any direction. So this is how the tumor suppressor gene acts. Now, one of the most important tumor suppressor gene is retinoblastoma. Retinoblastoma is an eye tumor. Is a tumor of eye. It occurs from the birth of usually a germline mutation occurs, and usually occur in the children of small age group. And it is famously known as governor of proliferation, this RB gene, right? So RB is a key negative regulator of the G1 and S phase, right? Synthesis phase of the cell. And it is inactivated in most of the human cancers. Now, this is how it acts. So, when you see that there is an growth inhibitors like the growth inhibitors actually stimulate the CDK P16, and the function of the P16 is to inactivate the cyclins. So, what happens is that when this pathway occurs, the cyclins are activated, and the cyclins keep a check on the cell cycle, and the RB stays in the hypophosphorylated state, that is, it is not being phosphorylated. The E2F remains attached to it, and whenever the E2F protein is attached to the RB, there will be no replication. There will be a transcription block, right? But if there are growth factors present and the growth inhibitors are absent, they activate they skip this inhibitor stage, and they directly activate the cyclin stage. Cyclin is the checkpoint, right? So the cyclins activate and causes phosphorylation of this RB protein, that is RB gene. Whenever there is phosphorylation of the RB gene, the E2F which is a protein which is being separated, and this E2F site goes and starts the S phase, that is synthesis and replication phase of the DNA, and it causes transcription activation. So this is how the RB acts. Now, retinoblastoma gene or RB gene occurs in 40% of the retinoblastomas, which are familial, and the mode of transmission is autosomal dominant. 60% occurs sporadically, and Knudsen has produced two-hit hypothesis of oncogenesis. Now, what is a two-hit hypothesis? You have to see this picture. This is taken from the Robbins, and this tells us that usually when there is a familial cancer case, when a child, when this is the female and this is the male germ cell, right? So the female is all right, but one of the gene of the male is mutated, and it is an RB gene, right? So it is transferred through the sperm into the offspring. One good healthy gene from the mother and one defective gene from the father. That causes a pair of a defective gene and a normal gene in all the cells, right? But still the cancer will not develop, right? And because one of the pair is fine. But what happens is that there is a second hit. There is an acquired mutation after the birth, and the two genes are then defective. The complete pair is defective. then it is known as the mutated gene, and this mutated gene that develops into the cancer or retinoblastoma. So this is the Knudsen's two-hit theory. The similar happens in the sporadic form. In sporadic form, both parents are alright. So the genes inherited are actually in the normal state, right? So when the offspring is born with normal genetic makeup. But what happens is when there is a mutation, further mutation occurs in the body, some of the cell may acquire single mutation. Later on, they may acquire the second hit. This is the first hit and the second hit. Second hit means the second mutation. When it occurs and both of the parts of the chromosome are mutated, then the cancer develops. Somatic retinoblastoma mutation observed in these tumors. Retinoblastoma, osteosarcoma, which is a tumor of the bone, glioblastoma, which is a tumor of the brain, small cell carcinoma of the lung, breast cancers, and bladder carcinomas. Now, that was the governor of genome. TP53 is a guardian of genome, and this is another very important gene that is being placed in our body to save us from many type of carcinogenic factors. And there are multiple functions of this gene also. So what it happens, it actually regulates the cell cycle progression. It just keeps on checking the cell cycle for any defect. Whenever it finds any defect, it just stops the cell cycle. It is a very powerful gene. So it can check for the DNA repair also. It can also check for the senescence, that is, complete stoppage of the cycle, and it also takes care of the apoptosis. So it has multiple functions. It's the most frequently mutated gene in human cancer. So DNA damage and chromosome abnormalities occur into P53 gene, and when there is an abnormality in the P53 gene, what happens that DNA repair could not occur, and normally the P53 gene, when there is a cellular damage, the P53 is in a normal condition, it causes cell cycle arrest. Then it causes the DNA to repair. If the repair is done nicely, then it will restart the cell cycle. And this is how there is genetic stability. Now the other process of P53 is that it causes apoptosis of the genes which are not being repaired properly. So when there is apoptosis, there is programmed cell death, and they are eliminated from the damaged cells are eliminated from the body, and it again causes genetic stability, right? Now, P53 can cause transient cell arrest, as I just explained to you in this picture, that it causes transient cell arrest and causes the DNA repair, right? It can also cause a senescence, that is a permanent cell arrest, or it can cause apoptosis. These are the three very important functions of the P53 mutation that you should know all the time when you are dealing with neoplasia chapter. Alright, so there is a Leigh-Fraumeni syndrome, and Leigh-Fraumeni syndrome is actually a syndrome in which the TP53 gene is mutated, right? So, and it is an inherited disorder. So there is an inheritance of the mutated TP53 in the offspring, and what it causes 25-fold greater chance of developing a malignant tumor by age 15 in the general population. So most of the tumor or the cancers that occur in the body have got this gene TP53 mutation, and some of the cases, the Leigh-Fraumeni syndrome, belong to the Leigh-Fraumeni syndrome. So I just explained to you the function of the P53 gene, but let us revise again. This is basically the normal cell in which the P53 is normal. It detects the DNA damage. It causes the repair of the DNA, and then whether it causes the cell cycle to stop, or it arrests the G1 phase and causes successful repair, or if repair fails, it causes through the Bax gene, it causes the apoptosis, right? But what happens when there is a mutation of the P53? When there is loss of P53 or mutation in the P53, DNA damage occurs, right? But it could not be detected, and that damaged cells, it goes into the cell cycle. There is no cell cycle arrest, no DNA repair, no senescence, and the cell remains mutated. When this mutant cells, it acquires the replicative activity, and it expands and multiply and becomes a malignant tumor. So mutated P53 with loss of P53 function, the DNA damage goes unrepaired, driver mutations accumulate, and of course there is malignant transformation. Now there's a list of other tumor suppressor genes besides RB and P53. You should just know the name. APC, E-cadherin, CDKN2A, TGF-beta pathway, P10, NF1, NF2, that is neurofibromatosis 1 and 2, and then WT1 pathway and patch1 pathway. So this was all about, I think this was more than, what you require to know at your stage. But I tried my best to summarize it so that, you remember. The cell cycle, we know that there are 4 phases of cell cycle, G1, S, G2 and mitosis. And we also know now that there are certain cell cycle regulator or checks which are being placed by the nature to check the any defect in the cell cycle, right? And then there are mutations. There are driver mutations which are cancer causing mutation. There are passenger mutations which do not cause cancer. And then there are multiple acquired mutations, right? And then there are gain of function mutation. The gain of function mutation means that there is proto-oncogene and oncogenes involved. And loss of function mutations are when there are tumor suppressor genes involved. I think this summarizes it all, and please concentrate. Go and read through the short book of Robbins, which will help you develop the concept. When the concept is developed, then you will be able to remember the points nicely. Please, I request you that if you have any query, if you could not understand any point,